Okay, yes, today is the 7th May 2020, and we have here with us Oskar Staufer. He's from Max-Planck Institute for Medical Research in Germany. So Oskar, as PhD student, he obtained a Bachelor of Science in 2015 and Master of Science in 2017 in Molecular Biotechnology from Heidelberg University as a German National Merit Foundation Fellow. So in his research, he applies the principles from biomaterial sciences and bottom-up synthetic biology to understand cell-cell signaling mechanism. So I hand it over to Oskar.
Thank you very much, Carolina, for this very nice introduction. I think you might have seen on the invitation, I also have a current kind of visiting position with the University of Bristol. You can imagine, currently it's not so much visiting, so it's not an active position now. This is where I want to start. I hope every one of you and your friends and family are fine wherever you are. If you stayed up late or if you woke up early for this talk, I hope you're doing fine. You're getting very good through this Corona time. And I think this is where I also need to thank Carolina, especially in general, for setting up this whole meeting, for putting lots of work into the meeting, making this a great opportunity, not only for the EV field to keep science going, to keep communicating, but also I think, and this I might say because I'm not so much an EV researcher, just recently got into the field, I might say this because it brings a lot of transparency to the field, so people from outside can see what are people in the EV field discussing about, what current problems do they see, how can they overcome these problems, and I think it's very valuable in general. So Carolina already said that I'm more or less a biomaterial scientist, even though I got trained as a biotechnologist. This is why the more or less first 10 slides of this presentation gives you a little bit information about my background, so you can see why the technology I and we in the Institute develop is actually maybe quite nice for the EV field in general. So what we are mostly doing as biomaterial scientists, as physicists, as chemists, as biologists in the department is something that we call bottom-up synthetic biology. Bottom-up synthetic biology I can explain to you in basically one slide. We see cells as biologists. We see, for example, such a time-lapse phase contrast movie of cells, how they adhere on a substrate, how they migrate, how they just live in general. And biology has been doing a great job, let's say in the last 40, 50 years, to develop tools how we can study, for example, this adhesion process as they came to our understanding down to the molecular level, what kind of architectures, molecular architectures you actually need in order to have a cell adhering to a substrate. We can build models out of this. And after a lot of research, what we will find is quite of a descriptive model, I would say, which is maybe complete at some points, incomplete at other points, but it's certainly not very intuitive. And we don't have so much understanding about the dynamical interactions, spatiotemporal interactions, and so on. So the general idea of bottom-up synthetic biology is to break down the complexity of these systems to a level that we can actually understand what is happening. How we do this? Well, basically we look at cells, like you can see living cells on the left side, and we think, can we build synthetic models that recapitulate some of the most central aspects of these cells, like you can see on the right, which is maybe for first instance just a lipid vesicle that should mimic the cell membrane of a cell, the compartment of a cell. I've especially been interested for many years now in understanding how cells communicate and so on. So this is where you will see how the whole thing of synthetic biology and synthetic EVs will relate to what I've been doing previously. But the general idea of bottom-up synthetic biology is that it strives to construct minimal lifelike materials, such as, for example, synthetic cells. Could be also other things, could be synthetic organelles, synthetic EVs, like I'm going to show you, whatever, by a bottom-up reconstitution to understand lifelike phenomena in general. We ask very fundamental questions in this field. We kind of ask, one does, or how does matter become alive? How do proteins, amino acids, nucleic acids get in touch with each other in terms of, or to actually build something that we consider as a living system? And this is what the talk will be referring also to. How do these materials that we construct that might have lifelike properties, how can they be integrated in actual living systems to form part of this living system? Just to give you a quick idea how synthetic, bottom-up synthetic biology looks like at the current stage where we are. This is an example that was published by our lab recently. It shows you synthetic cells in terms of compartments that have been filled with tubulin and kinesin molecules, ATP, and so on. And this is how we can study within these synthetic cells, spindle formation, cell division phenomena, and so on. As we are biophysicists, we're also, of course, interested in aspects like that relate to the cytoskeleton. So we are now at a stage where you can quite nicely build actin cortex in this synthetic cells. We can control this actin cortex. So this is how we see synthetic cells, and we try to put, step by step the molecular components of a cell into such a system and try to understand at each level, every time we integrate a new module into what does it do with the system. How I got into the field of extracellular vesicles, I was reading a little bit about vesicles. Of course, we use a lot of vesicles. And I saw that, I think especially the EV field has suffered a little bit from technical challenges, from poorly defined systems, from systems that have impurities in it, which has somehow impaired greater understanding of the EV-related phenomena. And this is where I thought, okay, maybe we can provide some help at least. Yeah, we can build very defined systems, very complex systems. Maybe with our technology and our knowledge, we can somehow provide a better defined system that EV researchers might even directly apply to understand their problem better, or as a control for their system or whatever. So the general idea of this talk, what I will present you, is how can we take not EVs from a biosource by isolation and end up with a, let's say, less defined system of a mixture of EVs, proteins, and so on. But how can we take, compared to this molecular components, lipids, nucleic acids, proteins, can be sugars, can be whatever, assemble them in vitro very precisely to give something that's a synthetic EV that will trigger somewhat equivalent response in the cell or in a living system. Why do we do this? Well, of course, first of all, you can imagine that this has some implications in clinical settings. Regulation agencies will be happy if you can provide a well-defined system with high reproducibility, no batch-to-batch variation. But I think it's also could develop into a very important tool for the EV research in general, because the defineness and the control over this synthetic EVs can uncover some fundamental mechanisms. And I hope I can show you at the end one example of how we applied them to at least get to the stage of proposing one of those mechanisms. So the problem, however, of this, as you can imagine, this is not like assembling a car, assembling a house, assembling something in the macromolecular world. This is engineering on the molecular level itself. So it's a very delicate and complicated engineering problem for the beginning. And this is why we need very sophisticated engineering approaches that allow us to actually use very fundamental physical phenomena or principles, thermodynamic principles, to construct these synthetic EVs in a controlled manner. So the next two slides will be dedicated to the systems that we use. I can already now say if I don't manage to explain this correctly, or if you cannot follow me, you don't need to be it's not important for the understanding of the rest of the talk, but I just want to give you the fundamentals of the techniques we use. One of those techniques is that we rely a lot on is the droplet-based microfluidics. Maybe some of you have heard about it. It's a very simple technique, actually. It relies on microfluidic chips, so basically small channels in the micrometer range where you can flow fluids through. We mostly apply aqueous fluids, which could be, for example, any kind of a PBS, cell culture medium. You can see here, these little dots are cells. And the second fluid we apply is oil fluids. And if we intersect these two fluid types at a T-junction or a cross-junction, we can get small droplets. This is the same technology that's more or less applied for single-cell sequencing analysis and so on. And the important thing about this is that the video is very much slowed down. Yes, we can generate hundreds to thousands of these droplets per second. So it's a very high throughput technique. But it does not only give us compartments that we could use as synthetic vesicle-like things, it gives us a whole plethora of different microfluidic modules that we can apply to manipulate these droplets. So to do sequential processes, so we can, for instance, mix these droplets to have a homogeneous solution inside. We can inject defined amount of fluids into these droplets. For example, if we have one process that should first proceed before we inject reagents for our next process, we can make two vesicles fuse so that we have a defined starting point of a reaction. I'm sorry. We can make these droplets small and small and small until we get something into the EV range of droplets. And what is most important at the end, whatever we have assembled within these droplets, we can also then release again into pure water-filled environment, so we can interface it with cells at the end. So this is how we envision the construction not only of cells, but also of synthetic EVs. We have a production pipeline basically with different models where we inject, assemble, construct step by step the individual components of the EVs. And I'm especially going to focus on lipids, on proteins, and on nucleic acids, on microRNAs. That's the general idea. How we achieve this, let's say, thermodynamic or electrostatic assembly of the vesicles, I will show in this slide. If I get confusing at some point, and I already put you the take-home message of this slide in the upper left corner, it's basically that this technique is a high throughput technique, which has a very exact and narrowly defined control, and it has an almost optimal encapsulation efficiency for microRNAs, so proteins and so on. So the general point where we start are these water and oil droplets, which are shown here. And into these droplets, we put RNAs, for example, microRNAs, and small lipid vesicles, liposomes, basically. So what will happen with these liposomes and the RNAs, they will diffuse within this droplet very fastly, of course, it's a small droplet. And at some point, they will encounter the periphery of the droplet. In the periphery, we have a surfactant layer, which we need to stabilize the droplets. And some of these surfactants are actually negatively charged. If we add magnesium into a solution, the magnesium will be dragged to this negative charge. The liposomes themselves actually, again, now negatively charged, which will drag them again to the magnesium. And as this electrostatic interaction is so strong, it will make the vesicles fuse and create a supported lipid bilayer on the periphery of the droplets. I can show you here in this movie how it looks like. We see here a droplet with lipids labeled in green, and you will see how we flush in this negative charge surfactant, and you can basically see the assembly of this lipid bilayer by seeing the lipids, how they get dragged to the periphery of the droplet and forming this vesicle. So this means we have a charge-mediated formation of a droplet stabilized vesicle, which has all the microRNAs and so on, or proteins inside. And this we can then, at the end, of course, release, as I said, into an aqueous environment to interface with cells. That's the magic trick behind the vesicles. And it's a very fast technique. So we can produce enough of these vesicles to treat mice, which is the most complex system we have interfaced the vesicles here, but also for human therapies or whatever, within seconds to minutes. So this is basically how some of our fully synthetic extracellular vesicles look like in a confocal microscopy image. You can see that we have a lipid bilayer with surface proteins attached to this lipid bilayer. In this case, it's tetraspanine proteins, and we have microRNAs inside. To give you an idea of how defined the system is, I'm showing here mass spec data of the lipids that we put into the vesicles. These are 9 lipids. We can do basically as many lipid types as you want. Yeah, the technique is very versatile. We just took these 9 lipids as they are the most important ones, as we saw from literature. In the left column, you can see how we designed the lipids ratio to be, which is closely resembling what we know from EVs, how the lipid ratio is. And on the right column, you can see the mass spec quantification data of how the lipid ratio in such vesicles are. And you can see it's almost perfectly matching of what we designed it to be. And most importantly, if we interface these synthetic extracellular vesicles with cells, we basically see very much the same interaction patterns. We see uptake of the vesicles, we see localization to the endosomes. At some point, we see localization to the lysosomes also. But the general behavior seems to be very similar to that of natural EVs. Some data to show you how pure the system is. I think the left image, many of you might know, it's a transmission electron microscopy image of the EVs from K562 cells. You see the EVs in these purified samples, but you see lots of other stuff swimming around and being deposited on the grid. On the right side, you can see our synthetic EVs, and you can not only see that the size is very homogeneous, but you can also see that we basically don't have any other protein components or whatever floating around and kind of making the system unpure. So we can also show on protein level by basically page analysis. We can see here in line 1 and 2, 2 different batches that we obtained from a commercial distributor. On 3 and 4, we used the very same cell line and did our own centrifugation, which we are not trained at all. So maybe you get better results with your EVs. This was basically the first trials we had to isolate EVs on our own. On the right side, you can see EVs that have been coupled with 4 proteins, simplicity. So you see that our synthetic EVs, the system is way better defined, and the batch-to-batch variation, if you just quantify the protein profile in these gels, you will see they're basically identical. So we can have a very, very high reproducibility from batch to batch. So how we envision this in the future is that maybe we can set up a platform that's like you know it from when you order your PCR primers or any DNA construct. You have a website with an EV creator where you can simply choose the lipids that you want to have. On UEVs, you can choose what kind of nucleic acids you have inside, and at the end, basically which proteins you want to have inside or outside on the membrane. You can choose the size of the SUVs, the EVs that you want to have. You can choose the charge, which medium you want to have it delivered by, and if there should be any synthetic component that you need for your analysis. This would be the dream of this technique, that is open to the community, and everyone can, for a very low price, obtain EVs, synthetic EVs, either for control or directly applied for the question that they're answering. So what I want to show now in the next, let's say, hopefully 20 minutes, and I try to be fast, is 2 stories. First of all, how we apply these fully synthetic vesicles for EV therapy. And the second is how we try to apply them to investigate some very fundamental signaling phenomena associated to the EVs. Let's start with the first one. The general thing we did here is we got into literature. We had a look, are there any known EVs that can be applied to therapy? We found this paper where people isolated fiber-side-derived exosomes, and they applied these for wound healing in a mice model. They described 3 very specific effects they showed in vitro, which is proliferation of epithelial cells, collagen deposition, and increased collective migration of epithelial cells. And they provided some data on the tetraspinin coding of the vesicles and on the microRNAs that are inside. And what we decided, and one reason why we chose the paper is, of course, because apparently they were so sure that their system works that they funded a company based on this, which has now been sold to Regenesis Therapeutics, which gave us additional confidence that let's try to see if we can reproduce the very same therapeutic effect they see with their EVs, natural EVs, in a synthetic EV system by just putting all the the components they described in their system into the synthetic EVs. We did the same characterizations. We started with epithelial proliferation and measured basically proliferation of keratinocytes by Higgs staining. And I think in this graph, you can already see that one of the fundamental advantages of this EV system, the breakdown in the complexity. We can step by step put in different tetraspanine, so TS here is a tetraspanine, different tetraspanine proteins one by one onto the membrane and measure which effect do the individual components have. And we see at the end that if we put all the tetraspanines combined, all of the three, we have the highest proliferation of the epithelial cells. We can then, again, step by step put in the different microRNAs they described. And we see at the end, if we assemble everything together, we have a quite good proliferation of these cells. So the first effect they described, the epithelial proliferation, we got checked. The next one we went on to is the collective migration of the epithelial cells. This you can assess by so-called in vitro migration models, where you basically culture epithelial monolayers with an exclusion insert, and then after they form a monolayer, you just remove the insert and you watch them basically closing this synthetic wound site in vitro. You measure what is the distance that the monolayers traveled by quantifying the free area, and you do basically the very same analysis again. You test tetraspanine by tetraspanine and microRNA by microRNA until you get a construct or a knowledge of how the individual components will influence the migratory behavior. And what is most important, if you're looking on the pure therapeutic application, you see these fully synthetic extracellular vesicles promote also the migration into the wound site by epithelial cells. And the collagen deposition, I'm now just going to skip. We got very similar results. It just get boring. It's always the same principle of tetraspanine by tetraspanine, microRNA by microRNA, but we also see increased collagen deposition of dermal fibroblasts that have been treated with these synthetic constructs. To put this on even a higher level, let's say in terms of therapy, we decided to go for a more humanized skin model, which is not just epithelial cells and cell culture, but we applied human organotypic skin models, which have or basically resemble very closely the architecture of human skin. You have the upper part, the stratum corneum, you have an epithelial layer, and you have a dermal layer. Below, you can wound these skins with, let's say, 3 millimeter wound sites, and then you can treat them with different either FSCVs or with the human serum in this case, and just quantify from this histological stainings how they close the wound, how efficiently. So you do by measuring how big is the wound, the remaining wound site, and how big is the thickness of the regenerated epithelium. And again, we can see that the FSCVs there have very, very good effect on the wound healing properties of this layer, which is comparable to the, let's say, commonly accepted positive control of human serum that for sure also contains exosomes and other EV types. So basically, we can conclude that these FSCVs might have a equivalent or at least very, very similar therapeutic potential as natural EVs have, but have the advantage of being very defined and very reproducible, very pure systems. We then went one step further and we said, okay, this is a good therapeutic potential, but we need to understand what are the components that trigger individual responses, individual phenotypic changes of these cells. And this is where we went to RNA sequencing transcriptome analysis. We compared keratinocyte cultures that have been treated either with PBS or with our full synthetic EVs. And we find quite a specific and very distinct change in the transcriptome pattern of these keratinocytes. We found 170 approximately differentially expressed genes. And then we made, I think, a very important control for this whole story is basically, and that shows again one of the advantages of the system. We treated the same cells with the very same concentration of the soluble components. So we just added the same concentration of tetraspanines and the same concentration of microRNAs. And in this case, we only find 5 differentially expressed genes, which are all also part of the genes that we find for the FSCVs. I think this tells us two very important things. First of all, that the FSEVs will induce a very specific response in the cells, but also that the soluble components are not as active as the EVs. And this might seem a trivial observation, but I don't think it is, because it's a very clear indication that we have something like a vesicle context. So you need to put the the components into a vesicle, onto a vesicle. You need to, if you want to see it in terms of information theory, you need to compress the information onto the vesicles so that these components can actually exploit their full signaling potential at the end. I will tackle this in the second part of the talk. We went further then to do pathway enrichment analysis to see which are actually the pathways that we trigger. You can see in the lower here in the lower part of this figure that it's mostly so these are the top 25 pathways that have been affected by the treatment. It's mostly pathways associated to adhesion, migration, ECM remodeling, and so on, but also pathways that somehow relate to proliferation, cell cycle progression, and so on, and many others. So it's totally, I think, over 70 pathways that have been significantly enriched in these FSEV-treated keratinocytes. We then combined this analysis with the advantage of the FSEVs, again, putting tetraspanine by tetraspanine, and then we can see which tetraspanine actually activates or regulates the expression of which gene. We can see which genes are overlapping, and we can again do, based on these data, pathway enrichment analysis, then see which tetraspanine combinations will induce which specific, let's say, general effects of the cells. Like, for instance, we can see that a combination of these two tetraspanines will mostly influence those processes that are somehow associated with immune regulation and inflammation, which makes sense. Of course, you want to have the immune system on the wound side to prevent you from any damage to or any infection at this site. You can dig into the data. You can see, for example, for this hemidesmosome assembly, which are structures that are very important for wound healing. Again, for every tetraspanine, which of the components or the proteins in such a hemidesmosome architecture is actually down or upregulated by which tetraspanine combination. And this is just to showcase you how you could apply the synthetic vesicles in order to get maybe a better understanding of which of the components in your EVs are actually triggering which response. We did the same thing for the microRNAs. I'm not going to show it at this point just for the sake of time, also because I started 10 minutes later. But it works in the very same concept. So what? Just to give a short summary about the therapeutic effects of these EVs, and we see that the ones we constructed have basically three major or can trigger three major processes in keratinocytes, which is migration, adhesion, ECM remodeling, and so on. The second one is everything that relates somehow to immune system activation. The third one is the cell cycle progression, which, at least in this model of wound healing EVs, makes a lot of sense and gives us a lot of confidence that the vesicles will have a good therapeutic response also in vivo. What did we learn from this? What kind of, I think, important observations would we have? Well, first of all, I think the information of this, the idea of this vesicle context is important. It's something that's cannot be done so easy with natural EVs, I would say. We definitely see that there are synergistic effects between the individual EV components. So we see that the whole is more than the sum of its parts, again, at this point. And I think most of these observations would have not been possible, or at least associated to lots and lots of work with natural EVs. So hopefully, and maybe we can discuss this later, and we can provide the technology here that helps you decipher the contribution of individual EV components to your system. So I would just show 10 more slides or so on the second part. So how can we apply these EVs to get some fundamental understanding of biophysical phenomena of EV signaling? How do they promote, how do EVs in general promote the signaling potential of the proteins that they have on the surface? And basically how we try to give a reason why cells use EVs at all to present proteins. And in order to understand this, we picked one system that maybe some of you are familiar with. It's a system of the cytotoxic immunological synapse. We choose this specific system because I think it's one of the examples where there's basically no discussion that EVs play a role. There's so much data, EM data, physiological data, other imaging data, that actually when a T cell encounters a cell that it wants to attack, it will form an immunological synapse, which is the process that you can see here. So basically a gap between these two cells. And we know from many, many studies that into the synapse, we have a directed release of vesicles. So this is for sure not a byproduct of any process that you can then find EVs in serum or whatever, but this is a directed release of vesicles from one cell to another cell to fulfill a specific function, which in this case is killing. Killing is achieved in this case by having a protein ligand, which is called Fas-L. You might know it as CD95 ligand. On the vesicle, these vesicles that have this Fas-L on their surface are released into the synapse and travel from one cell to the other to activate Fas receptors, which will then induce apoptosis. The general mechanism how this works, to give you a brief overview of what is Fas-L apoptotic signaling. So you have one or three Fas-L molecules. So currently people think that the Fas-L KDs is so high that you will mostly only find it in its trimerized form. It will then bind this trimerized Fas ligand, one of the receptors, which is shown in blue. It will add another receptor, which is on the cell membrane of the target cell, and then a third receptor. So you have complete Fas trimerization. So the Fas ligand serves as a trimerization site for the Fas receptor, which is then activated, but in terms of inducing apoptotic signaling, what will need to happen is that three of these activated Fas receptors will need to get into close contact, kind of a hexamerization event will need to take place in order to get this apoptotic signaling. The interesting thing here is this Fas-L is not only released on vesicles. You can also find soluble Fas-L. Because Fas-L itself has an intracellular domain, as you can see here, it's a transmembrane protein. But here in gray, these three arrows, these are MMP cleavage sites. And you can actually recover soluble Fas-L from serum or from blood, from patients, or from other fluids. And it has apparently the very same binding affinity to the receptor as the membrane-bound form. So all the important signaling parts of this protein are actually in the extracellular domain. And I think this is a very nice system to ask, when does a cell apply a soluble protein, and when does a cell apply basically a vesicle-bound protein? And interestingly, people have found that if this protein is on a vesicle, it's very active, and if it's not so active, even though the binding kinetics to the receptor is approximately the same. And these are basically the questions we asked and we wanted to follow with our synthetic system. So what we did is we got this very same Fas-L extracellular domain. We had it with a histag in order to immobilize it on the vesicles. And we treated Jurkat cells with either the soluble form of this protein or the protein that we attached on the surface, and we measured cell death or apoptosis by PI staining of the cells. And I think this is, again, one of the very, very interesting observations that we made, that you can take one and the same recombinant protein in this form. Yeah, it's the very same mixture. You can apply it to cells, you will see no effect. You put it on a vesicle, so it's not a biochemical change. You just immobilize it on the vesicle and you see it boosts by orders of magnitude its signaling potential. And this as it's such a hard effect, it should be a biophysical reason. It cannot be biochemical, very probably. We did tons of controls to verify this. We tested the vesicles, that they don't have a cytotoxic effect and so on. You can see here on the left just two examples. We actually assured that it's caspase mediated apoptosis. We observed our cells by time lapse in order to see that it's actually this apoptosis takes place when such a vesicle here in green touches a cell, but then this cell will undergo apoptosis, and it's not just because there are impurities on the system. So we are pretty sure that this effect is a true effect that only happens when the Fas-L is put on the vesicle membrane. We thought, how could this happen? Well, two things come into mind. First of all, there's a restriction of let's say, degrees of freedom when it comes to rotation, and between the soluble and the vesicle-bound form. And the second major difference is that the concentration, the local concentration, is high. We thought, okay, this might simply be can be attributed to local concentration effects. To test this, we built a diffusion model, mathematical diffusion model, where we basically built a room which is basically the size of an immunological synapse. We put in Fas-L vesicles in the soluble form, or Fas-L vesicles bound on vesicles, and we checked, which is shown here in green. What we counted is basically how efficient are these two forms in inducing Fas hexamerization. So we could count how many hexamerization events take place in this. What is good here is we can we can validate this model later experimentally, because we have a high control over the amount of ligands that are present on one vesicle. We can model this again, and we put in tons of other parameters into the model, all kind of physical parameters that will influence diffusion and binding kinetics between Fas and Fas-L. So if you have any questions on this, we can maybe discuss this later. What we observed actually at high local concentrations, so here we will see hexamerization events of the model and the Fas-L concentration. We saw that at high concentrations, actually the hexamerization is going down. Why is that? Basically, if you have not so much Fas-L, you will see nice trimerization. But if you have very high concentrations, you will see that one Fas-L will catch just one receptor, basically, and you will have no trimerization of the effectors. And this has been experimentally validated already by others. On the left side now, I'm just trying to hurry up to not steal too much of your time in the morning or in the afternoon, wherever you are. We can also see in the model that if we apply Fas ligand in the model in the form of a vesicle, you will see completely different hexamerization dynamics compared to the soluble form. And this we also saw in using our synthetic vesicles with different local concentrations on the EV surface. Why do we think this happens? And now we can just look at the model, what's happening. If you look in the soluble form, you will see that even if the Fas ligand is trimerized, this meeting of two trimerized Fas is just diffusion based. So you might have from time to time an apoptotic signaling. But for the big vesicles, at least, you will kind of sequester these activated receptors below the vesicle surface, and you will up concentrate by more and more receptors that just meet into below this vesicle. You will induce their interaction, or you will promote their interaction. And to test if this model is actually true, we can simply produce smaller vesicles, because the smaller the vesicles, the more pronounced this effect should be if the vesicle, if the Fas-L concentration on the vesicle remains constant. This we tested in the model. We see different kind of hexamerization events for different sizes of the vesicles, and we also see it in experiment. The smaller the vesicle, the higher the apoptotic signaling events, the higher the apoptosis rate or efficiency of these vesicles, which I think is a very interesting observation again. We also tested this. I'm going to skip this the explanation of this, but we tested it not only for Fas ligand but also for other ligands, RUNC, and we tested it for MHC molecules that also need to trigger TCR oligomerization. We see always this basically the same behavior, that the smaller the vesicle, the higher the signaling event. So basically what I think vesicles do is that they are a presentation platform that will drag any kind of things that will need to interact, either people like we can see in this image, or receptors on a very defined local spot, and vesicles will make these proteins interact at one specific spot at the end. So to sum this up, I think we kind of succeeded in constructing FSCVs that are inspired by a biological blueprint. We explored the therapeutic potential of these FSCVs, as well as that we, by using this transcriptome analysis, we kind of figured out the synergistic functionalities between the individual components. And in the second part of the story that I showed you, I think I demonstrated that immobilization of the ligands on the vesicle is very important, and that this concept of vesicle-induced receptor agglomeration could be a general mechanism that's present for many EVs, and that if this shows to be true, this Vera effect, then this is a very good biophysical reason why cells should apply EVs and not simply release proteins in the soluble form. So this is, I think it's a very good way to reason why cells need to apply EVs, which is maybe not so intuitive for many biologists in the end. So I need to thank several people for this. First of all, the 4 students in the upper left corner that helped me a lot with all the work. Julius did a lot of the work on the simulation of the things, and Jochen did a lot of work with the Fas ligand. I also need to thank my supervisors, Ilya Platzman and Joachim Spatz, as well as some funding agencies. And I think one of the persons I need to thank the most is actually Irina Nazarenko from Freiburg, who kind of assured me that these data that we obtained and the concept I developed is or can be important for the EV field, and who's given me a lot of support and kind of communicating this, for example, in this way with you guys, and try to help, at least from our biomaterial perspective, for your research. And I would be happy to discuss with you and anything that you find interesting in the data.
Okay. Thank you very much, Oskar. Very interesting talk. Can you stop the screen share? Okay, thanks. So I invite everyone to turn on their video, especially if you want to ask questions. And you know, that is really awesome, the way you can design the vesicles. And I guess all the tetraspanions, you can't really tell us what they are because of the nature of the patent, or you know, the development that you're doing.
I forgot to mention this at the end. I have a conflict of interest in this. Yeah, so we're trying to patent this technology, or there's a patent application currently. So I cannot yet disclose these proteins, but we are actually, because we're not experienced in the EV field, we're looking for people that can help us to shape these data in form of a manuscript somehow, so that we can publish it somewhere. Because we know how to write biomaterials papers, but we don't know so much how to write EV papers, and we don't want to add another paper that's just providing more confusion to the field.
I'm sure there's a lot of people who's interested, and but I have like one question that I really want to know. The answer is like, if you compare those tetraspanions, do you find any difference in terms of efficiency of uptake if you have only one tetraspanions compared to, you know, a variety of 3 tetraspanions on the vesicles?
So I can only answer this on a quantitative level, not so much on a qualitative level, because measuring uptake is not trivial, at least. Yeah, so you might have seen in the videos, most of our vesicles are fluorescently labeled by integrating fluorescent lipids. So we can measure how much of the vesicles are retained by cells, for example, after washing. And there we see a correlation, yes, that the tetraspanions will influence how many of the vesicles are retained by different kind of cells. But I cannot tell you if there's maybe any difference. Yeah? Some might be endocytosed faster, there might be a different intracellular sorting or whatever. This, we simply don't have the know-how on how to analyze the specific pathways.
Okay. Yes. Okay. That's very interesting. Okay, we have a few questions here. I'm going to read Muhammad questions, the first one, and then I'll let Aaron to ask the second question and combine it with his own question. Just because Muhammad has no microphone at the moment. So his first question is, with this, the mimetic or synthetic EVs, can you design them so that they have a naturally, or mimic the naturally presented surface receptor?
In terms, so if I understand correct, the question is how many of the surface receptors we can put and how natural these surface receptors are actually. So
mimic the natural.
Yes. We for the moment we use recombinant receptors that are just simply being purified from biological systems. Of course, you could argue this is kind of where the trick in the system is. We still rely on biological systems for at least for the proteins. But there are two things in this regard to say. First of all, the purification techniques for the proteins are way more sophisticated than I would say for EVs. So the purity of the proteins is higher. And we actually can also use just synthetic peptides that we can put on. If I saw correctly in the chat, I can take one of the other questions just into this answer, which is how we immobilize the proteins in there. And that's basically the trick. We can put transmembrane proteins in the right orientation. There are several techniques by attaching other protein moieties with this tetraspanine, for instance, that will guide the directional integration into the lipid layer. But what we found, I didn't show you this because of the time, is that for all these signaling, you will basically just need the extracellular domain 2 of the tetraspanines. This is simply extracellular, so what we got is his-tagged extracellular domain 2 of the tetraspanines, and then we just couple the his-tagged version to anti-A lipids. And this is yet living from living systems, but the extracellular domain 2 we could also just print in the peptide synthesizer and use completely synthetic ones.
Okay, Aaron, go ahead.
Thanks. Thanks for a really interesting talk, Oskar. So we were just wondering about how if you introduce your synthetic vesicles into an in vivo system, whether you have any ideas or thoughts on avoiding immune detection.
So there's we don't have a lot of experience with this. Yeah? The only thing we did yet is we injected the EVs intravenously in mice. And because these are synthetic constructs, the first thing we checked at is actually, do the mice survive the very first seconds and minutes? Yeah? Because it could be that there's agglomeration that they get strokes or heart attacks or whatever, which we didn't see. We observed the mice for 48 hours and they showed no apparent, let's say, behavioral changes. We didn't see any immunological, so they showed no apparent changes, but we didn't check in vitro or by any kind of immunological analysis if there's immune response to these EVs. We see that there's a certain degree of clearance from the mice circulation system, of course. But we have another project where we actually try to use these EVs to activate T cells, which gave us a hard time, at least. Yeah. So apparently the immune activation properties of the EVs themselves are not as good in vitro as we were hoping for in this project. So I can give you probably not answer at the moment.
Thank you.
Okay, Clotilde?
Well, thanks, but actually, I mean, it was very nice talk, but Oskar already answered my question about the membrane orientation of the proteins and the fact that you only use the second domain of tetraspanine actually makes a big difference. Thank you.
Yeah, but in general, it's not a problem to use the full membrane proteins. We published a paper where we used integrine proteins to put them into the membrane, the full integrine proteins that are functional also at the end, and ATP synthase and whatever. It just complicates the system more than what you will need for a therapeutic response, maybe. if you're just interested in a response, then you can stay with the extracellular domain too.
Yeah.
Okay. Thank you, Clotilde and Oskar. Sujin. Hi, Sujin.
Okay, hello. Thank you. Very nice technology. I wonder if the lipid composition in UV is critical for the FS-UV formation. For example, the content of phosphatidylinocytol in FS-UV is very low, and what would happen for the FS-UV formation without it? I wonder this about. Thank you.
So I think this is one of the big advantages of our technologies compared to other liposome production technologies, electroformation production. There are several out there. We don't rely so much on the lipid composition and on the buffer composition that we're using. The only thing we know, or the only thing we need, more or less, is a slight charge. It can be either positive charge, net charge of the vesicle, or negative charge. Negative is usually more biological relevant. There are almost no positively charged lipid membranes in biological systems. But as long as the vesicles have a slight negative charge, we are good to go. Then we can basically insert any kind of molecule, any kind of lipid, I'm sorry. And we did this also with lipids that have incredibly large head groups, so pegylated head groups or RGD modified head groups, so peptides on the head group. So then we are very versatile with the system, and we can integrate large amounts of different or many kind of different lipids at the end.
Okay. The next question, I'm going to read it because Maya doesn't have a microphone. So about the post-translational modification of the surface protein. So specifically glycosylation, if you use that synthetic protein, you assemble it into the synthetic EV. Can you comment on that?
So I guess many people know better than me that glycosylation of the tetraspaminase is very important for the signaling function. And this is, of course, something that you would need to take into account when applying synthetic peptides. There are ways to integrate glycosylated amino acids into such a synthetic peptide. They're not so straightforward at the end, but it's possible to do this. And I think, maybe correct me if I'm wrong, the glycosylation patterns and sites of the extracellular domain 2 at least are very well known for most of the tetraspaminase. So in this case, we can mimic this again in a very synthetic system. So far, I cannot show you any data, any comparative data between the natural and the synthetic ones, because simply it's not so conclusive at the moment, what we got there. But in general, this is something how you could overcome this limitation.
Okay. Yeah. Thanks so much, Oskar. It's very promising technology and it's very exciting. I'd like to thank you for keep going and give us a very nice presentation. Even though we have that internet difficulties at the beginning, I think you deliver it really well. So thanks so much for that.